Okay, so hello everybody and welcome back to part 2. I accidentally did a wrong presentation, so here's the new one. The one of the cohesion protein shows sheets and helices. And so let me show you the back. So here's atoms at exercise. Here are the color codings. Here are the color codings. Side chains on the protein. We see few hydrophobic side chains on the surface. Because the wave view shows a hydrophobic core inside the protein. Carbon's shown as gray. Cutaway view showing how amino side chains. Here's another view. Close up. You can notice there's like tyrosine, tryptophan everywhere. Different angles. Here is it. Is always visible. Notice the surface of the ring of hydrophobic. Then seek out the protein's core causes the protein to fold, giving rise to the beta sheet and hydrogen helix hydrogen bonding patterns. Here, as you can clearly see. So here, that's different renderings. Protein with structures or unstructured peptide make up the protein. Are shown. Cohesion binds bacterial enzymes together is much, and another shown in green that dense plant material. Amylase is an enzyme found in the cell intended unless to digest that a starch molecule about to be cut up by the enzyme. Trypsin is responsible for digesting proteins. Here, you see forming yellow as it digests a peptide. Hemoglobin is a red blood cell. It contains 4 hemes, orange, which bind oxygen. Hemoglobin delivers oxygen from and launch to all the other cells in our body. Rhodopsin is found in rod and cone cells in the eye's retina. Rhodopsin contains a small molecule called retinol, and purple which absorbs light. The protein gives us our vision. P53, guardian angel of the genome, very famous of the oncoprotein. We'll talk about this in the primal assignments. Keratin is is found throughout the body. It's famous for making veganized fish and reptiles give feathers and horns. Myosin, the muscle protein. This structure is part of myosin, which is one of the makeup muscle fibers. As the protein allows to move our limbs. I'll talk about it in episode 35. That's a long time. Green fluorescent protein, or GFP, found in the two globular muscular or jellyfish find one another. Look then at the see if you can, as the hydrogen has been removed to make viewing the images easier. GFP folds into a barrel structure. It's known as a beta barrel. Protein folds is directed by amino acids pointing either into or out of the barrel. Here's a cutaway view. Pointed into the barrel, pointed out of the barrel. Now here's complex forms found in the mitochondria and heterozyte cells. This is because the protein needs it to function. ATP is this famous molecule with the energy loop produces ATP. Now ATP, I'll talk about it as a nucleotide. It's our energy source, adenosine triphosphate. But I don't want to spoil it, so I'm going to save that for later. Branch, misfolded disease causing fruit. Because the proteins for reasons that are not fully understood. This is a protein and cause diseases like mad cow disease. But these are beautiful, they're like patterns, but they're kind of messy. They're oxygen revolving complex. Photosynthesis, chlorophyll, and beta-carotene molecules, as well as many other specialized molecules, have this protein use the energy from sunlight to move electrons from water molecules. And RNS. They're like everywhere. Okay, I was doing a little too much. Protein diet. Why do we need protein diet? The protein we eat is made of amino acids. We have to break it down to amino. When we then eat proteins, we cannot eat proteins from another amino. For example, if we ate green for jellyfish, we would not glow. The protein would simply be broken in digestive systems, but the main liquid both the proteins could be used to make you remember. Might be more accurate was a protein on the nutrition facts tables to say amino acids. cysteine has a special role in protein folding. It's known as a disulfide bond. I think I talked about this before, or at least made a proof. Linking two parts of positional bond called the disulfide bond helps fold the protein. Insulin is a hormone and controls how our body processes sugar. People who do not make enough insulin have diabetes and must inject insulin to live. Insulin protein folding. Insulin is a small protein made of two peptides shown here in pink and blue. It's a pretty small thing. Insulin has a small hydrophobic, so does not fold well without disulfide bonds. But however, 3 disulfide bonds are needed to help insulin fold properly. This one links the two peptides, keeping them connected. This one links part of the same peptide, helping to fold the protein. This one links the two peptides near the end of the pink peptide A. Counting the cysteine carbons helps us see which cysteines form disulfide bonds. Red and gray cysteines form bonds inside the same peptide, while the others form between peptides. So here is just a close-up image of it. Insulin is the same in other animals. Proteins are remarkably similar in all of these things. A pig's insulin, look at the amino acid sequence. So a human is the same thing. Give all of this, everything's the same except at the very end that T and A, and it's not a problem. Humans have serine and pigs have an alanine. It's the same. Look, give Q C C T S I C S O. You'll notice it. That's how pig insulin can be given to humans with diabetes. This has saved millions of lives. But so are flies. Even flies has new mammal insulin. The amino acid sequence is quite different, but there are many similarities. Like there's extra part, and this is, but it's the disulfide bond also. For insulin between flies and mammals because both have the similar pattern of hydrophilic amino acids and the same disulfides. Mammalian and fly. Notice how many acids are the same in mammal and insect insulin. And here are the answers. The flash sequence skips the R. If you insert the dash, we see that even more amino match between the mammal and insect sequences. E, D, and N are similar in size or charge. L, V, and A are similar in size and are hydrophobic. They are so similar. Interesting, isn't it? S and Y both have alcohols in their side chains. And so, according to 78, there are 8 similar amino acids shared between mammalian and fly insulin. This makes 25 matches between mammal and fly insulin, but 15% of the amino acids match. So cool, right? That share similar hydrophobic or hydrophilic properties at each sequence position. Counting similarity history means that mammal and fly insulin are 70% the same. Now we can see the fold the same even with different sequences. We can clearly see. Other members might be considered similar to one another. You now know a lot about proteins. And now is the final end. We'll see you later, and bye for now. Don't forget to subscribe.